This video is going to continue the exploration of our central dogma of biology with transcription. And so for this, we have a few more learning objectives, again, focus on that central dogma, the steps of transcription, again, what happens in prokaryotes versus eukaryotes, the sequences, the proteins, all of the things involved, as well as the steps for RNA processing in eukaryotes that happens after transcription, but before translation. As a reminder, here is our central dogma that describes the flow of information from DNA to RNA, eventually to proteins. Now keep in mind that there are exceptions to the central dogma. Like viruses like HIV, retroviruses can actually do reverse transcription and go from RNA to DNA. But here we're going to focus on the basic process of transcription, going from DNA to RNA. Now, transcription is going to involve 5 basic steps: RNA polymerase binding, initiation, elongation, termination, and in eukaryotes, we have a 5th step, which is processing. For prokaryotes, they don't have a nucleus. So as soon as that RNA is made, it can start to work with ribosomes to make the polypeptide in the translation process. But for eukaryotes, the process of transcription occurs in the nucleus, and the process of translation occurs at ribosomes, either in the cytoplasm or those on the rough ER, if you remember from our module about organelles. And so this is separated processes in the eukaryotes. To start, transcription uses an RNA polymerase. So do not mix up RNA polymerase with DNA polymerase. Here we're talking about RNA polymerase. And in bacteria, there's only one RNA polymerase. And it's got an interesting structure. We call the main part of it a holoenzyme. So it's got 2 alpha subunits, 2 beta subunits, and then this sigma part. And if there's no sigma, then it's just the core enzyme. So with the sigma, it's a holoenzyme. Without the sigma, it's a core enzyme. And what sigma does is it's going to recognize a sequence on the DNA and allow the RNA polymerase to come in and interact. What I'm showing you here is the promoter region of a DNA. So if this is our start point, our start sequence, upstream of that is going to be the promoter. And there are key residues at the -10 and the -35 spot that have consensus sequences that are recognized by the RNA polymerase, and even further upstream is where the sigma region is going to bind. We call the first A in that in our coding strand, or the first T in our template strand, the +1 site. An initiation in bacteria is going to involve some scrunching. So that sigma region is going to bind to our up element upstream of our promoter region, where the alpha beta parts are going to bind to those consensus regions at -10 and -35. And the polymerase will start to synthesize a strand of RNA using complementary base pairing to the DNA sequence. However, it'll do something called abortive synthesis, where it moves forward and then backwards and forwards and backwards, and it creates this extra amount of mRNA, this scrunching. And it will do this until it gets enough energy to escape the promoter and to begin elongation. And elongation is going to read the DNA in a 3' to 5' direction and create the RNA in a 5' to 3' direction. And this, unlike DNA polymerase, is de novo synthesis. So there are no primers involved. It can just bind and go. And remember, in the language of RNA, there are no T's. Instead, we use U's or uracil. Now, RNA polymerase is able to do some backtracking to help with proofreading, so it can reverse and move forward to help with that error detection. And elongation will continue until it reaches the termination region. For prokaryotes, that can be of 2 flavors. The first is when it reaches a region that has GC rich, and those G's and C's, when that RNA is made, will start to interact with each other and will form something called a hairpin loop. And the hairpin loop will help dislodge the RNA polymerase. RNA polymerase can also use something called a Rho factor if there's no hairpin loop. That will again aid with termination. And keep in mind, for prokaryotes, there is coupled translation. So as this mRNA is being synthesized, we can then have ribosomes come in and start translation right away. For eukaryotes, transcription is more complicated. First, eukaryotes use 3 different kinds of polymerase, depending on what kind of RNA is being made. So RNA polymerase 2 is the one we're going to focus on because this is the one that makes pre-mRNA. And depending on what polymerase is used, the promoter region can also be different. So there are some characteristic elements for RNA polymerase II here. We've got a TATA box, a DPE site that's downstream of the transcriptional start site. We've got these other regions upstream of the transcriptional start site. This is forming our core promoter. Keep in mind, for other polymerases, we can have even more upstream control elements, all the way up to -80, and there can be enhancers and other regulatory sites, both upstream and downstream of the transcriptional start site, that +1 site. So this is overall a more complicated promoter. Now, elongation is going to come, but we first need to get initiation. And initiation requires, for eukaryotes, transcription factors. And they're going to form a complex with each other, so there can be more than one transcription factor that are going to bind to that core promoter. And what is really important for this is they're going to have protein-protein interactions. So there's going to be interactions between multiple regulatory transcription factors at the core promoter, at other sites, and then eventually interacting with the polymerase in order to get the initiation required for transcription. Once it's transcribed, then it's going to need to be processed. So shown here is a pre-ribosomal RNA, and their processing is for all sorts of RNA. So it's whether it's ribosomal, transfer RNA, or messenger RNA, all of them for eukaryotes are going to involve some sort of processing step. Transfer RNAs are also processed again. They have modifications, removal, and then eventually you're going to go from this pre or primary transcript into your mature tRNA. So the mRNA are going to gain some special modifications here. So we're going to first add a 5' cap. We're going to add a 7-methyl guanosine cap to the 5' end. And what this does is it helps increase the stability of our mRNA. Because the mRNA has to travel, it's got to go from the nucleus into the cytoplasm, it can be degraded by an enzyme called nucleases. And so by adding the cap, it helps slow down that degradation by nucleases. This cap, as we'll see in a little bit, also helps position our mRNA in the ribosome. Now, not only is it capped, but the pre-mRNA for eukaryotes is spliced. And what this means is that it's processed so that parts are removed and the remaining parts are put together. So the language we use is we're going to remove regions called introns, which are intragenetic regions, and we're going to keep our exons, which are expressed. Exons are expressed, and we will eventually put those together. And this is usually done by a complex of enzymes called a spliceosome. And the interesting thing about splicing in eukaryotes is that we can have alternative splicing. So if we see here our pre-mRNA where we've got 2 introns and some exons here, we can have different combinations, right? So we can make both just the regions on the red, or we can have the region with green in the middle in our final mRNA. So this creates some variety from a single pre-mRNA product. This is the spliceosome in action, so it's going to recognize a few characteristic sequences, a GU, and then an A with a small space, and then an AG. And so the spliceosome complex will come together, and it will snip and remove that intron, leaving behind the exons, which will then be joined together. And that excised region will very quickly be degraded. Now, I said that there was a 5' cap. The 3' end also gets a modification for eukaryotes. So the 3' end gets something called a poly-A tail, which is just a whole bunch of A's. And it's added by an enzyme called poly-A polymerase, which uses ATP to add on that region. And this functions similar to the 5' cap. It protects it from nuclease attack on this end. And it also helps the mRNA get recognized for nuclear export. So it helps this transcript leave the nucleus to the cytoplasm where the action is going to happen. And that's going to be where our next process is, translation.